We will begin around 10 or so. The online edition, something we usually do in person over 2 intense weeks. But for this year, due to COVID-19, we've brought it into an 8-week sort of online experience and expanded it to everybody who could come. We've had an overwhelming response. Over 3,000 people have registered, which is truly amazing to me, from all over the world. And we're really excited to be able to talk to you guys. The goal of our school is to introduce you to what we think are some of the most exciting areas of physics, in particular, the bridges and the links between physics and living systems, sort of broadly defined. We are trying to gear the course toward undergraduate students studying physics. We welcome anyone else to come along, but that's our sort of intended audience when we've prepared these lectures and these discussion sections. We want to show you how elements of what living systems do connect to things you've learned in physics classes. We want to introduce you to new subjects in physics and biology that are of interest to physicists. And in doing so, we prepared a very broad range of topics to tell you that span from the smallest molecular and cellular scale things in living systems all the way up to what brains and animals and groups of animals do together. And so in some sense, our goals are as much inspirational and aspirational as they are educational. We want you to learn something, but we also want you to get excited about new things that you might not have seen. So all of the attendees are muted and cannot have their video on. We just have too many people to have a truly interactive experience. And so the way you can interact with us is through the question and answer. If you see something in there that you like, please upvote it. We will see the top upvoted questions, and that will let us figure out what topics or questions are common to more than one person. Also, these lectures will all be recorded, and we will upload them to our YouTube channel. That may take us a few days, so you don't need to worry about recording them. They will be recorded. So I'm Josh Shavitz. I'm a professor of physics in the Louis Sigler Institute for Integrated Genomics at Princeton University. I'm also one of the co-directors of the NSF-funded Physics Frontier Center for the Physics of Biological Function. And I'll be giving today's lecture in the afternoon. I also wanted to introduce Bill Bialik, the other co-director of the CBBF.
Introduce as in you expect me to say something.
Yeah.
So yes, let me add my welcome. Let me say that one of the things that we've enjoyed about doing this in person is really getting to know students from a wide range of backgrounds and from all over. That's obviously going to be much more difficult here. Our view of you will be through the questions that you ask. On the other hand, there are already, let's see, 4, 6, there are already 25 times as many of you as we can accommodate when we bring all of you to one place. And so we're hoping that the spirit of being able to have a dialogue with people from around the world will outweigh the cost of not being able to do it in person. It's where we all are now. As Josh said, the goal is to expose you to the things that we're most excited about and that we hope you will find exciting as well. At a moment when it can influence your choices about what you do next, and also just provide you with some basis for further exploration. So we really all take this limited question and answer bandwidth very seriously. And so we hope that you'll put as much of your energy into that as you can. And I will stop because there's plenty to do today, and we'll each get a chance later. So thank you.
Okay, I think we're going to let our 3 discussion leaders, Ben, Bratton, Endao, Han, and Deanna, take it from here. And so I think Bill and I and Halima will turn off our videos, and I hope you enjoy the discussion section.
Okay, so the three of us have put together a couple of slides. So most of you were able to send in some questions in advance. So I really love the fact that everyone is greeting us from all around the world. I think some of these greetings from around the world we may push to the dismiss category just so we can keep the science questions floating to the top. But we're so glad to see all of you. And we prepared some answers, but we'll just have to see how things are going as we go along. Deanna and Endao and I are all students and postdocs. So I'm a postdoc, Deanna's a grad student, Endao's another postdoc who work with Josh. So we think a lot about the bacterial and microbial world. And so we've kind of divided this Q&A session into 3 parts just to facilitate whose screens are being shared. And so that's going to be, I'm going to talk about the sort of biological aspects of microbiology, then Deanna is going to talk about microscopy, how we see this small world, and then Endao is going to talk in generally about micro swimmers and things about fluid mechanics that may be relevant for today. As Josh said, one of the main questions that came up was this sort of general idea of what kind of background do people need? And the answer is some. We have no idea exactly what your background is, but we expect you to have some background generally in physics or thinking about how to use math to represent physical phenomenon. And hopefully you have seen some type of biology at some point, just a little bit. You have some idea what biomolecules are. So when we talk about things like proteins or DNA or genes, things like that, that resonates somewhat. That being said, at least the 3 of us, and as you will hear from Josh and Bill later, our email addresses are available to you. So if there are other questions that come up later, you can definitely send us emails or find us on Twitter. You know, we will somehow we reached you, and so somehow you can reach back to us as well if your questions don't get answered today. Deanna and Endao, do you have anything else you want to add in this sort of opening section? Okay. So there were a ton of questions that are super interesting, that are very relevant for modern microbiology, modern microbial biophysics, including questions about what happens when you have multiple species interacting, what happens when you have cheaters that come in, how can they take advantage of the situation, questions that have to do with spatial game theory and sort of spatial organization of things. These are super great questions. We're going to try to provide you some resources. We're going to, in the same, maybe the same location, maybe a different location from where the papers were posted for you. These question and answer slides will also be available, maybe with some other links. So we're going to try to direct you to some resources there. We just don't have enough time to cover all of those today, including other questions about the bacterial cytoskeleton. That's something that I'm particularly interested. That's where my specific research focuses on. The main thing to know is that for the relevance today, that most bacteria have a rigid cell wall. That is one of the load-bearing structures. They also have extracellular appendages that can be quite rigid and flexible, that can bear the mechanical load of the structure. And then finally, bacteria are super relevant for things like hospital-acquired infections, that bacteria can live on implant surfaces, that bacteria can live in flow. All of these things are quite important. We just don't have time to cover them today. What hopefully we can cover a little bit about is some of the nomenclature that has ended up being confusing throughout the years. So swarm is a very general term, and not every swarm is the same. So swarm, thinking about swarm motility, it's really like a verb. It's how bacteria swarm. This is different than, say, Colobacter swarmer cells. This is different than the swarm plates or swarm assays that you may have seen. It's different than like a swarm of mosquitoes or a swarm of gnats. And that can be really confusing. You know, in physics, it tends to be that if you can write down the math equation and everybody can read the math, then they agree on what it is. You know, maybe there's some terminology or symbology that's a little different. But in sort of general spoken science and in biology, there can be words that have loaded meanings that are just not quite conveyed entirely. So that's also very true when we think about the flagellum. Some of you may think of this in sort of historical terms, like the weapon, the whip. Some of you may think of eukaryotic flagellum that is shown here. You know, this is a large bundle of filaments that bends and beats. For today, what we're interested in is the prokaryotic flagellum, which actually has a slightly different structure. It's rather smaller than the eukaryotic flagellum, and it uses ATP, and it burns ATP, this molecular sort of energy molecule of the cell. It burns it to spin this large molecular machine. So in bacteria, people tend to name proteins with a four-letter combination. So you can see here we have like FLIC or FLID, and this choice comes from you name it based on some kind of function. That's usually like the first 2 or 3 letters, and then it just gets an extra letter. So that means each protein encoded by the genome has a unique letter that is the name of that protein. And now this can be a little bit confusing because sometimes people pronounce the letters individually, like FLIC, and sometimes people try to read it out like it's an English word, like fly-C. And so this is something that, you know, can be confusing as you're reading or you're listening to people talk about things, knowing exactly what protein it is. And really the cell is like, the way to think of it is the state of the cell is a list of all of the concentrations and locations and post-translational modifications of all of the proteins in the cell, right? So bacterial cells have, let's say, 4,000 or so genes. And so at any one point in time, it's like a 4,000 dimensional vector. You know, there's post-translational modification. So maybe it's like a 10,000 dimensional vector that has the concentrations of each of the proteins. And so keeping track of all of that is, of course, very difficult. And that's why people try to collapse this and do dimensionality reduction on whatever their particular system of interest is. And so how this can go is within there's a lot of signaling that goes on. Let me see if this video will play. So some of these signaling molecules or signaling pathways are how you take input signals and you use the sort of standard molecular biology of gene regulation to change the amount of RNA and the amount of proteins in the cell. And so these are things like transcription factors. These are things like 2-component signaling that come about. And that's generally how you, in a non-spatial sense, have the flow of information in the cell. Now, there's also reaction diffusion patterns that get set up where the action of one protein changes the behavior of another protein, for example, in this MIN system, which for some reason is not looping. But I think you can see that as the chemistry in the cell, it spatially localizes the chemistry in there. And so that leads to a spatial flow of information. Now, when you're thinking about between cells, there's also a sort of genetic flow of information. I would say that that's a little bit harder to think about, but really there's spatial flow of information between cells. So for things like chemotaxis or phototaxis, anything that ends in, you know, TAXIS is about cells moving toward or away from a signal. So you can have gravitaxis, where things follow gravity. So plants either, you know, they send their roots down, they send their leaves up. Flies have negative gravitaxis, they tend to fly upward, things like that. All of that is about sensing some signal, and that signal is some type of molecule or signal where you're able to take either a temporal or a spatial derivative. And so we'll see a lot, especially in chemotaxis, where you somehow try to trade off between the spatial derivatives and the temporal derivatives. And therefore, you're able to measure, are you going up a gradient or are you going down a gradient by making 2 measurements at different points in time, correlating that with space, or at 2 different points in space and correlating that with time. Finally, there are also molecules that allow the cells. So in that sense, I think of chemotaxis and gravitaxis as the cells are executing a plan, but they still need to signal, they still need to send information based on how that plan is working out. However, sometimes cells get to a high density or they get to a low density and they actually have to change what they're doing. And so a common stereotype for this type of signaling is called quorum sensing, where cells secrete a sensor molecule and they take that molecule back in and they're able to measure the local concentration of it And that's the way they vote and sort of say, do we have enough cells around? And so when that happens, what they're able to do is then change through their gene regulation pathways what molecules are being produced. So there's a question from Max, it looks like, about the time frame we speak of when we're thinking about information transport and signaling in the cells. So this information transfer can be very fast. It can be definitely on the sort of millisecond time scale. And that is sort of the chemistry level of things. You can change the phosphorylation state of proteins very rapidly, and then that can lead to them moving about or changing their signals. Also, things like ions flowing in and out of the cell, that happens on the second or sub-second time scale. And it can go all the way in bacteria out to many hours or many cell generations. For example, if you are thinking of cells that have to change from being a filamentous phenotype to then be dividing, or if you go and you decide to sporulate. So if you're in bad conditions and you want to sporulate. So there's really a lot of different types of timescales that are relevant. Okay, let me just quick read a couple more of these questions. It looks like a lot of, thanks everyone for sending in the questions and for everyone else who's helping answer these as we go along. Okay, so yes, it's definitely true that sort of single cell decision making and like, do you need neurons to make decisions? That can be a hotly debated topic, especially in groups where, you know, some of us study single bacterial cells and others study neurons and collections of neurons in brains. I would say that this is some type of decision making that based on some set of information, the cell ends up in a certain state. That I would consider that decision making. Okay, another set of questions is about what types of scenarios can these motilities, different types of motilities, be relevant in? And the answer is, it's really hard to replay history. So when we think about evolution and why these things evolved a certain way, the best we can do, really, in my opinion, is find a situation where such a motility would have an advantage. So I picked one example here where this is, you can imagine you have a bunch of flow lines. We have channels here, and the cells, some of the cells are able to move upstream and some are only able to be pushed downstream. And so this is a situation where if you're able to do both, then you're able to colonize these other branches. So it's a somewhat artificial environment that it happens in, but it's very reminiscent of the flow that happens, say, in plant leaves or in the human vasculature. So if you can find a condition where you might have a benefit and that's not so obscure, you know, it's not like I took these two things and only when certain astronomical phenomena are happening and it's like an even day of the week, then you get a benefit. You know, I think that that's a reasonable way if you have something like that. That being said, evolution definitely also happens on microbial timescales. So this is an image that apparently did not reproduce very well for Zoom purposes, but I'll have a link to this paper. So this is the Lenski long-term evolution experiment, and this was in 2012. So it started in 1988. 12 flasks of bacteria, and the Lenski lab continues this evolution experiment to this day. There's a brief shutdown for COVID-associated reasons, but you know, when you can get to tens of thousands of generations, you can really study what happens in these kind of long-term evolution experiments. Okay, and then I think my last set of questions was just about how can you tell the difference between like a raft or a swarm or swarming cells. You know, this is a difficult thing to decide what the best way to talk about this is. I think of a raft as the set of cells that are doing the verb swarm, and then that means that they are swarming cells. So I don't think there is a swarm that's a noun typically used in this context. And when the cells come together and exhibit these kinds of rafts, or they are able to swarm, it's a somewhat operational definition, right? It's a type of motility that tends to be flagella dependent and doesn't happen in single cells. So if a single cell can do it, then it's probably not swarming. And you know, I think this is an open area of research about whether, like, what is the difference between swimming compressed into 2 dimensions and in a dense cellular environment, and how is that different from swarming? And so people have tried to scrape up cells and do things like transcriptomics, measure that state, right, that 10,000 dimensional vector that is the state of the cell. They've tried to measure that in cells that are swarming, as traditionally defined, or swimming as traditionally defined. And they do see differences in what proteins are expressed, how many of them, what RNAs are there, things like that. So it seems like there is a difference between swarming and swimming, like compressed swimming, but we're still actively looking at that. Okay. I am going to stop sharing my screen while I read some more of these questions and see if there's some more of them that I can answer as Deanne is getting ready. Well, I'm going to attempt to stop sharing my screen.
Hi, everyone. Can you see the right screen? Not the presenter view?
No, wrong one.
Oh, wrong one. Okay. So now you see the right screen?
Yes.
Okay.
Yeah.
So a lot of the questions we got from you guys were about how are these motility experiments actually done in the lab, or what are the experimental techniques that are used to observe this moving bacteria. And so we thought that it might be interesting for you to learn a little bit about microscopy. So microscopy is one of the cornerstones of biophysics. And if you read a biophysics paper, almost certainly you will find something about we use microscopy to do this thing or other. And so we thought it would be interesting for you to understand a little bit more about this. So in general terms, what is a microscope? A microscope is something that allows you to see something really small by magnifying it. And you can say that in general, it consists of a light source, some sort of sample, and a detector, and these are connected by beam paths. And so your light source can be lots of things. In the case of optical and electron microscopy, it can be, or your source, sorry, I said light, but also for electron microscopy, you can have the sun or a candle, you can have LEDs, you can have lamps, you can have lasers, or you can have electron guns. And then the detector is something that is gonna detect this, whatever you're emitting from the source. So it's gonna be something like your eyes if you're looking through an eyepiece, or a camera, or it can be film if you're developing into a film, or it could be a point detector if you're kind of doing like a scanning of your sample. And then we go to our sample, and this is what we're interested in studying. But it turns out that for biological samples, well, most of the things we're interested in are actually transparent, they're very small, and so it's very hard to distinguish what you're interested from respect to the background. And so there is a lot of different techniques that are used to increase that contrast between what you're interested and what you're not interested in. So you could be looking at like the kind of more intuitive things to think about, like say is light being absorbed, or maybe I'm looking at the reflection of light from the surface. But you could also go into other physical properties of light that we know about, such as, for example, light has the waves of light have a certain phase, and we could use that somehow. Or we could talk about, oh, this material is kind of birefringent, and that's going to affect light in some way. Or we could try to target a specific parts of the cell by using fluorescent tags. And so in this next slide, I have in the left side of the screen 4 images and videos of tardigrades or water bears. And I actually took these images 2 years ago in a summer school in microscopy. So something similar to this, not overseen. And so this is meant to illustrate these different contrast techniques. Which contrast technique you choose depends a lot on what are you trying to look at. So in the top left, you can see a bright field image. And the principle of bright field image is similar to that of like having those hand shows that you do where you have a candle or a lamp, you have your hand, and then on the other side, you have a screen. And you're projecting some sort of shape on the screen, and people are like, oh, it's a bunny or whatever. And so what you're seeing is just the shadow of whatever you're imaging. And so that's how absorption works. We have our light, our sample, and then we're imaging from the other side. And so the parts of light that are being absorbed will look up dark on the screen when we're imaging. In the case of dark field microscopy, it's exactly the opposite. So we have, it's kind of similar when you're wearing a watch and there is the sunlight and it hits your watch and it bounces off and it sometimes hits someone in the face or something. You're looking at that reflected light from the face of your watch. So in the case of dark field, you're looking at scattering. So you have your imaging source, you have your sample, whatever it is, and then you're imaging from the same side as your light is coming from. And so what you're seeing is just the light that bounces back from your sample. So that's why it's in the dark background, because we're not seeing any of the background light. And in this left corner, we have one of these advanced techniques that I mentioned. This one is called DCI, or differential interference contrast. And DCI basically measures 2 regions of the sample that are very close to each other, and it kind of tries to look at differences in the path lengths, optical path lengths. And one thing that is kind of important for you to know about DCI is that it's very sensitive to having birefringence in your material. So currently, a lot of the slides or the places where or like the sample prep that you use can be plastic, and plastic is birefringent. So if you ever think about doing DCI, bear in mind that you have to be a little bit careful about this kind of details. And I would highly recommend that you go and read about DCI because it's super interesting how actually you produce your image. And then in the bottom right here, you have a polarization image. And the idea is that water bears are actually birefringent. And so when you image them using polarized light, the orthogonal components of the light will have different optical path lengths. And so you're able to reconstruct an image just based on the fact that your water bear is birefringent. Another use for polarization microscopy is you could have uniform thickness samples, and then you could see what are the different components that make up the sample. So those are the 4 images that I have of tardigrades. As you can see, you get different information based on which contrast technique you're using. On the other hand, we have fluorescence microscopy, and this is one of the most used techniques for biology studies. And in this image, this is one of the cells that I study. It's an E. coli cell, and you can see that it's happily swimming around. And inside the cell, I have little particles that are labeled with green fluorescent protein. Normally, when I'm doing my experiments, my cells are fixed, so they're not moving around like this one. And I try to measure what is, how are these little particles inside the cell actually moving. For the purpose of this demonstration, we wanted to show you a swimming cell. So next, what is the simplest microscope possible? This is like the basic building block of any microscope. And it consists of 2 lenses. Usually one of them is called the objective and the other one is called the tube lens. And you place your object right at the focus of the first lens, and you place whatever imaging device you have at the focus of the other lens right here. And the idea is that when you have light that is coming from the focus and it passes through the lens, it will continue traveling in a parallel fashion until infinity. If you now take another lens, when it goes through that lens, it will focus down at the focus. So basically, this is a way of taking the light from this small arrow to this big arrow. And as you can see, there is some magnification involved, and that magnification is given by the ratio of the two focal lengths of the lenses. This kind of system is called a 4F system because, as you can see, there's 4 focal lengths. So you have your 2 lenses are separated by the sum of the 2 focus, and then you have your sample and your object at the focus of each lens. And when you go and you buy an objective, usually it says like 100x or 20x or 50x. And what that means is that if you use it with the right tube lens, as is specified by the manufacturer, then that ratio f2 over f1 is going to be 100 or 50 or 20. And what is really nice about putting your object at the focus of the lens is that it's a lot easier to set up your samples and things like that. So in practice, the objectives are several lenses, and they're designed such that as long as you're within the working distance of the objective, it's as if your whatever your sample is always at the object plane. And so this is the way that most modern microscopes work. In the past, they used to, the sample wasn't at the object plane, and this led to lots of complications, but now most of the microscopes work this way. And so you can use this kind of 4F setup to build more complicated things. So one of the things that we do in the Shavitz lab is that we build our own custom instruments. And so here you can see the instrument that both Ben and I use for our research. And this is a home-built microscope. And so what you can see is that here in this region over here, you have that 4F system that I just showed you. This is our objective, and this is our tube lens. And next, you can add additional things to your path. So for example, here we have a filter wheel that allows us to filter out different wavelengths that we're not interested in. And here we have another pair of lenses. So this is another one of those 4F systems, and this gives us another additional magnification of 2x. And so in total, our microscope has a magnification of 200x. And I wanted to go back to that thing that I told you at the beginning, that a microscope is your source, your sample, your detector, and the beam path that communicates them. And so here in this rectangle on the left, we have our source. And in our case, we use lasers. So we have 3 different lasers. And then this corresponds to this area over here in this image. And you can see that there's lots of optics. But the idea is that it's just what all the lenses and mirrors and different optics that we need to get that light to our sample. Then here we have our microscope body where we place our samples. And usually your objective lives there. And this is the corresponding microscope body in our setup. So we place our sample over here. It's hard to see because the condenser is in the way. And we use micrometers and a little piezo stage to move things around. And then in the rectangle on the right, the blue one, you see our detection path. And as you can see, we use an EMCCD camera that corresponds to this camera over here. And again, all these lenses are just there to provide the magnification, the filtering, and well, the filter wheel, and all the things to get that light into that detector that we're interested in. So although an optical table can look a little daunting at first, just remember that a microscope is just your source, your sample, and your detection system. And what is really, why would you want to build your own built instruments? Well, there's like 3 main reasons why you would want to do this. One of them is it's a lot cheaper to build your own microscope than to buy one straight out of the box. But not only it's a lot cheaper, but microscopes that are bought commercially usually are like a multiple function device. So you can do lots of different things with it. And so they're not specialized for one test in particular. But if say that you always know that you're going to image things moving inside cells, then maybe you want to tune your instrument such that it's specific for that purpose in particular. And so it has a very good resolution, or it has a very small working distance because you know that your bacteria are small, or it's very stable, for example. And those kind of things, you can really achieve them by using a custom built instrument. And the third reason why you would like to build your own instrument is that you have full control of your path of light. And so you can do interesting things to it. So for example, in our case, for my research, we have this little biplane module and rectangular face mask that we add just in the path of our beam path. And this allows us to take 2 images of the same focal plane simultaneously. So when you go into custom built instruments, you can add a lot of extra functionality to your instrument, and you control everything about the path of the light. And with that, I'm going to pass the torch to Endao, who's going to talk a little bit about fluid dynamics.
Deanna, as you're transitioning over, there's a couple questions maybe that you can answer about the limit of the total magnification possible. So in light microscopy or in any type of microscopy, what's the largest, or in that case smallest? Like what limits the resolution?
Okay, so what limits the resolution? Resolution is limited by your wavelength and by the numerical aperture of your objective. And it's basically just not, your objective is capable of capturing a cone of light. And so like the light that is coming from like at very oblique angles won't be able to make it into the objective. This is like a very simple explanation. And so you lose like frequency components in your Fourier space, and that leads to you not being able to have infinite resolution. So in general, it's more or less lambda halves, so your wavelength by 2. That's a good thumb rule. And then what is your max magnification? I'm not sure what is the limit. Ben, do you have an intuition for that?
Yeah, I mean, I think it just, so like for light microscopy, I think it is usually dominated by that type of, you know, the sort of single molecule emitter resolution sorts of things. If you think about other types of microscopies, it might be for electron microscopy that electrons are really hard to focus. So like the technologies for focusing light with optics, right? Like lenses work really well for light. Phase plates don't work quite as well for electrons. So I think it just depends on what type of modality you're looking at, what the sort of resolution limiting factor is.
Okay, should I start?
Sounds good.
Okay, can everyone see the right screen and can hear me?
I think so. I can hear you and the screen looks correct.
Great. Okay, hello everyone. So I'm gonna mostly focus on this paper written by Purcell about swimmers at low Reynolds number. By reading your questions, we categorize them, basically put them into these 4 questions. What are Navier-Stokes equations? What is Reynolds number? And how to swim at low Reynolds number, and how to outrun diffusion. So today I'm gonna briefly go through these questions. So firstly, Navier-Stokes equations is called equations, which means it has more than one part, right? So actually, these equations have 2 parts. So the first one is says the fluid is incompressible, and that gives you this divergence of velocity field is 0. And then the other one is basically another form of F equals ma, but we need to rewrite it so it works for a continuum. So for a single object, if there's a force F applied on this object, you know, F equals ma will describe its motion. But if now we're looking at an element in a bigger material, this object is has forces applied by external field. For example, its own gravity, or if it's a magnetic field when the material is magnetic. There's an external force, but also it also experiences forces applied by its neighbors, by these other boxes around it. So to describe the motion of the material, we need to define a velocity field, which is basically the velocity of every point in space x, y, and z, and also v is a function of time. Okay, so to describe the motion of a fluid, we need to define a velocity field. And how this velocity field moves is still described by F equals ma, but we need to write it in another form. So we are more interested in the local properties. So instead of using mass, we use density. And instead of using the force, we use a force density or something like that, which we call a little f. So now ma equals f becomes rho a equals little f. And the acceleration, there's also some change in the acceleration. So now, as you remember, the velocity is a function of position and time. So to calculate the total derivative of velocity, in this case, it's called the material derivative. We need to consider not only time, but also the locations x, y, and z. So since dx over dt is vx, dy over dt is vy, and dz over dt is vz, we can write this then in a more concise form, which is this. So that gives you the acceleration when you're considering the motion of a fluid. So now rho a equals little f becomes this form. So on the left, we have the inertia term, which is related to acceleration of the material. And on the right, these are just little forces from different sources. For example, the first one is the body force is from its own gravity, its own weight. The second one is from the pressure, and the third one is from the viscosity of the liquid. So due to the limitation of time, we can't explain every single term. But when you see this equation, you know that on the left is basically rho a, and on the right it's different forces. Okay, so this is a really complicated equation to solve. Actually, there's a $1 million prize related to solving this equation. So normally we consider simpler forms to make life easier. For example, in this case, normally we consider a flow that develops slowly, which means it's in a quasi-steady flow. So we can ignore this partial v partial t term, and also if you write this p term in a different way, you can actually absorb this body force. So we ignore that term, and then what's left is an equation like this. Now the pink part is the inertia term, which is related to acceleration and motion, the inertia of the fluid, and the blue term is a viscous term that's directly related to the viscosity mu of the fluid. So the Reynolds number is defined as the ratio between these two terms. So it's a rough estimation of which regime your liquid is in, right? So it's the order of magnitude of the inertia term over the order of magnitude of the viscous term. So if you estimate how the flow flows, how the fluid flows, you can always find a characteristic velocity of the flow, u, big U, and also a characteristic length scale of the flow, big L. So the inertia term is roughly rho u squared over L, and then the viscous term is roughly mu u over L squared. So if you do the calculation, you find that the Reynolds number is rho u L over mu. And this tells you the ratio between these two terms. So if we're in a regime where a Reynolds number is much larger than 1, it means the inertia of the liquid dominates. But if we are in a regime where the Reynolds number is much smaller 1, it means smaller than 1, it means the viscosity dominates, and inertia doesn't play any role in in the problem. Okay, so so in the limit of very small Reynolds number, we can even get rid of this term. So now this equation only has 2 terms left, and that's what we want. So now with all these simplification, the Navier-Stokes equation become these 2 linear equations. And there's a very interesting symmetry in these equations, and that's this one, right? So if you imagine you add a minus sign in front of the pressure. So if the pressure becomes minus P, then the solution, the new solution, is just minus V. Okay, so you can add a minus sign here, add a minus sign here, everything's the same. This means if you reverse the way you push the fluid, then you reverse the flow completely. And if you do it right, it actually seems like you are reversing time. So that's the cool part, right? So one pretty good demonstration of that is this experiment. So you can put a very viscous liquid in between 2 concentric cylinders, and you put a blob of dye in the liquid. And now you can make the inner cylinder rotates. And this system is also at low Reynolds number. And what happens is that if you rotate the inner cylinder, the fluid will be sheared to flow. So these 3 blobs of dye will be spreaded all over the place, right? So you can see these colorful ribbons that's in the whole container. But if you wait patiently, after several cycles, these people will turn the inner cylinder backwards to the opposite direction, and then that's when the magic happens. Okay, so I will just let this video play. I think they have one more cycle. Yeah, there. Okay, now they are going backwards.
Yeah, we were saying how this is one of the coolest experimental videos we have seen. It's just incredible.
Thank you, Diana. That's correct. Yeah, almost there. All right, now everything goes back, right? So if you do this right, reverse, the idea is if you reverse the way you push the fluid, then you reverse the flow itself, and it seems to be a reversing of time. So this is directly related to how a swimmer should move at low Reynolds number. So there are 2 consequences of these equations. The first one is the rate does not matter anymore. So for example, Purcell used a scallop as an example in the paper. He said, if you're a scallop, no matter how fast you close and how fast how slow you open, it doesn't matter anymore. So yeah, and the second consequence is that to move forward, the body deformation need to be non-reciprocal, which means you can take a video of the swimmer and then you play the video backwards. If these 2 videos look the same, then it doesn't go anywhere. They need to be different. So if you want to know the more details, this paper is a pretty good one to start with more rigorous proof in mathematics. But now today we're just gonna show you the idea how it works, right? For example, if you are a scallop or something like a scallop, there's only 1 degree of freedom, right? So the swimmer has 2 rigid parts and a hinge that connects them. Okay, it can close and then it can open up, right? So if you play this video backwards, it looks exactly the same. And also the rate doesn't matter, right? So if you close fast or open slowly, you'll go back to exactly where you are. And then since this motion is also non-reciprocal, it doesn't go anywhere, okay? To get a non-reciprocal motion, you need at least 2 degrees of freedom. For example, this 3-link swimmer that Purcell suggested in his paper. So it has 3 rigid parts and 2 hinges. So if you make the move in this cycle, in this sequence, you'll see, firstly you bend this one down, and then you bend the other one down, and you bend this one up, you bend the other one up. Now, if you play the video backwards, these two sequences don't look the same anymore. So this is a non-reciprocal motion, and then the swimmer can go somewhere. And one of the questions many people are interested in is which is the direction of motion. There's a simple way to see that, and the method is you're looking for a body wave created by the swimmer, and then the direction of local motion is in the opposite direction of the body wave. For example, if I have at least this works for cylinder bodies. Okay, for example, if you have a fiber that's waved on one end, and it generates this sinusoidal wave, and you can see now the wave propagates from the dashed line to the solid line, which is propagating to the right. So if you put this thing in a liquid at low Reynolds number, it will swim to the left. Okay, and the same thing is here for these swimmers. So you can. It's not rigorous, but you can kind of think of these as some waves. Let's see. You can kind of think of this dance as some kind of waves, and then and these waves, they propagate to the right. Right. So these waves they propagate to the right. So the direction of motion of this swimmer will be to the left. But this is not rigorous. When action moves, it will also rotate and all that. But this is a simple way to find out which way a swimmer might go. Okay. Okay. Now get rid of all these. Okay. The last problem is how to outrun diffusion, because bacteria live in a very noisy environment, right? So when when things are small, everything's doing random walks, right? So these, if you consider a swimmer surrounded by a lot of molecules, these molecules are all moving around all the time, right? So to know whether the situation is good or not, the swimmer needs to measure its environment, right? So, and the good thing to do is to measure something here, and then you go somewhere else and you measure it again, and then you compare your measurement from these two different locations, right? And then that leads to informed decision. However, since all these molecules are moving around, if you don't outrun the diffusion, then your two measurements will be highly correlated, which means you spend a lot of effort measuring the same thing, which leads to bad decisions. So the idea is we need to run faster or outrun the diffusion. Okay, so the length scale for diffusion is proportional to d, which is the diffusion constant, and time, the square root of d and time. And if the swimmer moves ballistically, the distance it goes across is the speed of the swimmer u multiplied by time t. So in this case, we want ls to be larger than ld, which leads to a time that's larger than big D over u squared, which means we need to go further than big D over u to have a reasonable measurement of the environment. Okay, this tells us. So this is why Purcell has a sentence in the paper. He said, if you don't swim that far, you haven't gone anywhere. This is what it means. And also it tells us it doesn't really matter how floaty you swim, right? As long as you go, if you swim slowly, you need to go further. But if you go really far, you can still outrun the diffusion. Okay, and yeah, these are the slides. And where is fruit? Yeah. So these are the emails of the three of us. So feel free to send us emails and ask more questions. We'll try to answer them as much as we can. Thank you. I think I'll leave this slide here. Okay.
So we've received a lot of questions in the chat, and I think we've all been trying to answer them there. So if you have, if you ask some things, check check to see if we have answered them yet, and we're still working on answering some of the others. And there was one about, oh, I lost it.
Oh, there are so many questions. Okay, which one should I start? Yeah, it's a Navier-Stokes equation solvable at all. Yeah, I think the answer is people don't know. People don't even know if it's solvable or not. Okay. Does that relate to reciprocal motions in the paper? Diana, which is the question you talked about?
I was looking for it, but I think maybe it got answered. I cannot find it. There is this one, the Purcell paper says that if a bug is in a liquid and if the viscosity is increased, then bug does not feel any change. Please explain this. It is written on the first page of the paper.
Yeah, I don't know what he's talking about. I think that the bug should feel different. Maybe he means that the dynamics of the flow is the same, but I think it should definitely feel harder to push the liquid, right, with the same rate. If you're pushing with the same rate, then you feel more resistance if the liquid becomes more viscous. Yeah.
Oh, there is one here that says, where did it go? Why do we talk about length and not about velocity when we talk about outrun diffusion?
Yes. So the idea is, yeah. So if you the diffusion length scale goes proportional to the square root of time, and the swimmer, if it goes in a straight line, the distance it goes across is proportional to time. So it doesn't really matter how fast you go, right? So you can go very slowly, but if you keep pushing, at some point you'll outrun diffusion, right? So that's why we have this relation, right? So if you swim really fast, you don't need to go that far for outrun diffusion. But if you go slowly, you need to go across a longer distance. But anyway, you'll still be able to overrun diffusion. Does that sound right, Ben?
I think so.
Yeah.
Yeah.
Yeah, that's a good question.
And maybe answering, there has been a lot of questions about how do you bridge the gap between going from being maybe a physicist into doing biology, or the other way around. And so I think that people are interested in what kind of resources would you use. So I can speak of the part of going from physics to biology, which is what I did. And for me, what was most helpful was a combination of everything. Reading books, going to some basic classes, but reading papers. But also one thing is going to biology talks. And at first you're gonna go and you're gonna sit there and you're gonna be like, I don't understand anything, what's happening? But after some time, magically you will start understanding what's happening. And it's not very clear to me how that happens, but it's just like being consistent and just like accepting that you come from a different background and that you want to learn how the other side speaks. And it's really important to think, oh, to not think like, oh, we're physicists, we don't care about what the way the biologists think. Really, biophysics is about a bunch of different disciplines all coming together and everyone working together. And I don't know if someone has a comment of how to go the other way around, from being a pure biologist to more like the quantitative aspect.
Yeah, I don't think we have any of those people on this particular panel, but
yeah.
Okay, so I think we have answered a lot. For the sake of time, I think we may need to try to answer some more of these. I think there's a way that we can save these questions, and we might be able to post more of the answers with the slides that we're going to put up for these Q&A. And also, we may be able to group some of them together and pass them along to Josh as he gives the lecture this afternoon.
Okay, well, thanks, guys. That was a successful first session. There were a number of questions about getting the size of the shared screens to work. There were also answers being typed in. So maybe look in the answered questions if you are still having trouble seeing some of the shared slides. It seems that many people figured out that we're having problems. Someone also started a chat group through groupme.com. It's in the question and answer up near the top of the upvoted ones. We did not start this. One of the users has started this, but you're welcome to use that. We will look into starting a Slack channel for this. I'm not sure what's involved in all of that. But we'll send out an email if we get something more official set up through Slack. Thank you all for coming, and I will see everybody in a little less than an hour for the main lecture. Okay. Thank you.